All right, and welcome to lecture number 3, where we talk about the production of antibody and we talk about the role of T cells and B cells. It's this cooperation that results in the production of antibody. T cells, or T lymphocytes, and plasma cells, or B cell lymphocytes, together recognize foreign antigens. They directly destroy some cells and or go on to produce antibody in plasma cells. There are 3 parts involved in the production of antibody: the macrophage part, the T cell part, and the B cell part. Step 1 is the role of macrophages in the production of antibody, and you're going to want to know all of these steps. The role of macrophages. The macrophage processes the antigen by phagocytosis and degradation. Meanwhile, it selects portions of the antigen and conserves them to present them to T helper cells. Interleukin-1 is secreted by the macrophage, which activates T helper cells that were previously resting. Step 2 in the production of antibody is called T cell sensitization. The T helper cell binds to the antigen in the macrophage, and this stimulates the production of lymphokines. The main lymphokine secreted is interleukin-2, and that is secreted by mostly T helper cells and to some degree natural killer cells. Interleukin-2 initiates proliferation of activated T cells. Activated T cells produce many T helper cells. The cells are suppressor and cytotoxic like memory cells. Step 3 in the making of antibody is B cell activation. Once T helper cells are sensitized, B cells bind to the T helper cell and the macrophage and the antigen. This tricellular complex of antigen, macrophage, and T helper cell activates the B cell, which generates receptors for B cell growth factor and B cell differentiation factor. B cell produce growth factor and make plasma cells and memory cells, while B cell differentiation factor makes the individual immunoglobulin classes. The final step in the production of antibody is step 4, and that's the termination of antibody production. Antibody production is terminated by the T suppressor cells, also known as the T regulator cells. Those actually downregulate the production of antibody. Let's go ahead and talk about the categories of immunologically active lymphocytes. We've got the B cells, and they're called B cells because these cells originate from the bone marrow. They represent about 15 to 20% of all lymphocytes, and they turn into plasma cells, and those plasma cells make immunoglobulins. We have our T cells, which originate from the thymus, and they represent 60 to 80% of all lymphocytes, and they are responsible for cellular immunity. Then we've got our natural killer cells. They represent about 5 to 10% of lymphocytes, and they secrete interleukin-2. We've got the lymphokine activated cells, and then we have the tumor infiltrating cells. Cells and viruses that are targeted from natural killer cells and LAK cells would be tumor cells, fetal cells, normal bone marrow and thymus cells, the hepatitis virus, and cytomegalovirus. Let's talk about the types of T cells. We've got what's known as the CD4 subset and the CD8 subset, and that stands for clustered differentiation. They are phenotypically similar to one another, so they are in the same subset. We've got our T helper cells. They regulate immunoglobulin production and activate B cells. And then also in the CD4 subset, we have the T delayed type hypersensitivity cells. They secrete lymphokines, and they are involved in type 3 hypersensitivity reactions. Then we have the CD8 cells. We have the T cytotoxic cells. Those are also known as T effector cells, and they destroy virally infected cells, tumors, fungi, and they are involved with transplant rejection. And then finally, the last cell of the CD8 subset would be the T suppressor cells, also known as the T regulator cells. They downregulate T and B cells and control the ratio of antibody classes. So just to summarize once again, here are T cells. We've got our T helper cells that regulate immunoglobulin production and affect B cells. We've got our T delayed type hypersensitivity cells. They secrete lymphokines, and those are both part of the CD4 subset. And then part of the CD8 subset are the T effector cells and T regulator cells. The T cytotoxic effector cells destroy virally infected cells, and they are involved in transplant rejection, while the T suppressor / regulator cells downregulate the ratio of antibody classes. The T helper cells are part of the CD4 subset. They aid in B cell maturation in the bone marrow, and this is overseen by B cell growth factor. They supply activating signals to B cells. They activate the generation of antibody classes, and this is overseen by B cell differentiation factor. And they also activate effector T cells, or those T cytotoxic cells. T helper cells are essential in B cell antibody class switching. We've got the delayed type hypersensitivity T cells, and these are part of the CD4 subset. They secrete macrophage chemotaxin and migration inhibition factor. They secrete transfer factor, and that makes the T cells more responsive. And they secrete interferons. The primary function of the delayed type hypersensitivity T cells is to secrete lymphokines, and these are chemicals that affect other cells. The T effector cells, or T cytotoxic killer cells, are part of the CD8 subset. They provide immunologic surveillance for malignant cells. They eradicate viral and fungal infections and also intracellular bacterial infections. They destroy parasites, and they play a role in the rejection of transplanted tissue. The suppressor T cells are part of the CD8 subset. They control inflammation produced by T cells. They control antibody production. They balance the ratio of immunoglobulin classes, and they suppress B cell response. The primary function of the T suppressor cells is to downregulate the actions of other T and B cells. Let's go ahead and look at our T cell surface markers, and these are typically proteins on the surface of cells that help to identify and enumerate the lymphocyte subsets. They can be used to predict outcomes of certain diseases like leukemias. We can identify these cells with what's known as flow cytometry, and this is a method that looks at cell size, shape, and density through what's known as scattergrams. So we have our T11 surface marker. This is called the erythrocyte receptor, and it is associated with female cervical tissue abnormalities. We have the T3 subset, or T3 surface marker, and that is useful for immunohistological tissue analysis. And then on the right, we've got our T4 and T8 markers. Those are common markers for immunodeficiencies. And then we have our CD25 marker, which is a marker for autoimmune diseases. Then we've got our interleukins secreted by certain cells. We've got our lymphokines and monokines. Our lymphokines are produced by T cells. And the lymphokine is a chemical released by T lymphocytes. When coming in contact with specific antigens, they stimulate monocytes and macrophages. Interleukin 2 is probably the most important lymphokine. Then we've got our monokines, which are produced by macrophages. A chemical released by macrophages that affects the growth and activity of other white cells is interleukin 1, and interleukin 1 is probably the most important monokine. So on this slide, I have a nice list of lymphokines and monokines, and there are some common properties of all cytokines. They are secreted in rapid bursts in response to cellular activation. They bind to specific membrane receptors on target cells. They act on different cell types, and they influence the actions of other cytokines. On the left, we've got our lymphokines, which are secreted by T lymphocytes, and on the right, we have our monokines, which are secreted by macrophages. And you can just take a look at those items on the list there. Let's go ahead and talk about the more specifics of interleukin 1. Interleukin 1 is released during inflammation. It is a potent mediator of inflammation. Interleukin 1 activates the T helper cells to produce interleukin 2. Interleukin 1 activates B cells, producing plasma cells, then antibody. Interleukin-1 will increase the oxygen metabolism in neutrophils, and during the phagocytic process, specific granules and lysozyme are released. This is all part of the phagocytic process. There are three primary actions of interleukin-1. Its actions on lymphocytes, its actions on inflammatory events, and its actions on the central nervous system. So the first action is its actions on lymphocytes. It induces interleukin-2 production. It influences suppressor cell activity, promotes proliferation and differentiation of B cells, and enhances natural killer cell activity. Action number 2 is interleukin-1's action on inflammatory events. It mobilizes neutrophils from bone marrow into the circulation. It attracts neutrophils, lymphocytes, and monocytes to affected sites by disease. It increases the breakdown of proteins and stimulates hepatic production of CRP, C-reactive protein, which is an acute phase reactant. And then finally, interleukin-1 reduces hepatic synthesis of albumin. Action number 3 of interleukin-1 is the actions on the central nervous system. Interleukin-1 can lead to an elevated body temperature. It can depress your appetite, and it can promote slow-wave sleep. Interleukin-2, also known as T cell growth factor, is a lymphokine secreted by T helper cells and to some degree natural killer cells. It generates the T cytotoxic cells. It's actually essential in T cell generation. Interleukin-2 promotes T helper cell function. It modulates suppressor cell function, enhances natural killer cell activity, and regulates interferon synthesis. Interleukin-2 is instrumental in the body's natural response to microbial infection and plays a role in antibody production. We don't spend a lot of time on the other interleukins, but I did want to mention interleukin-3 and interleukin-5. Interleukin-3 recruits new cells to mature in the bone marrow. It essentially replaces cells that have already been used up. Interleukin-5 stimulates eosinophil activation and aids in the switch to IgA secretion. It aids in the synthesis of the monomer IgA to dimer secretory IgA. Okay, so we already discussed the interleukins, and now I want to get into the interferons, which is a group of cytokines first discovered in virally infected cells. They inhibit viral replication, and that's where the name interferon comes from. They interfere with viral replication. Interferons also inhibit division of malignant cells. They inhibit cell division of T suppressor and T cytotoxic cells. They enhance phagocytic activity of monocytes. And they enhance activity of natural killer cells. Interferons most benefit patients with non-Hodgkin's lymphoma, metastatic breast cancer, multiple myeloma, and other miscellaneous cancers and leukemias. Let's take a look at our central lymphoid organs, or our primary lymphoid organs, and this is where our B cells and T cells originate from. Bone marrow contains precursor cells of lymphocytes that later mature in the thymus. B cells originate in the bone marrow. The bone marrow contains precursor stem cells of the lymphocytes that later mature in the thymus or bursa. The thymus is a flat, bilobed organ situated below the thyroid gland along the neck that expands into the thoracic cage. T cells originate in the thymus. They are usually fully developed at birth and contain numerous T cells. The lymphatic system is dedicated to the circulation and production of lymphocytes. Some functions in the lymphatic system are to transport immune cells to and from the lymph nodes to bone, and to transport antigen-presenting cells to the lymph nodes where immune response is stimulated. Mature lymphocytes are in the secondary lymphoid tissues and are essential in immune response. The lymph nodes contain both B and T cells. This is complex cellular stations along the lymphatic ducts. Those are your lymph nodes. The spleen aids in phagocytosis of blood-borne particles. Mostly B cells are in the spleen, and it aids in phagocytosis and immunoglobulin synthesis. The tonsils are a secondary lymphoid tissue. Most lymphocytes here are B cells. In fact, about 50% of the cells in the tonsils are B cells. And then in the gut or the gut-associated lymphoid tissues, this includes the appendix, we have B and T cells. Macrophages are the most chemically active of all the phagocytic cells and are important in antigen processing. So how do macrophages accomplish this antigen processing? They cooperate with B and T cells in the production of antibodies. They secrete components of the complement system. They secrete hydrolytic enzymes and secrete toxic forms of oxygen that assists in phagocytosis. And they secrete monokines that activates the T cells. There are a number of ways to identify cells. The first one is morphology, and that's the appearance of a cell's size, shape, and staining characteristics. And we can use flow cytometry to look at cell morphology. We can use genetic probing, which is DNA sequencing, or PCR, which is a molecular method for identifying cells. We can look at antigenic markers. That's basically looking at protein markers on the surface of cells. We can look at products secreted into the surrounding cells, or looking at cells' chemical secretions like cytokines. And then we can look at a cell's functional capacities, look at the functions of certain cells. Identifying cells help us to better treat and identify A-gamma globulonemias, HIV, and leukemias. All right, so that's a wrap for Unit 3. We'll talk to you soon with Unit 4.